Hello students, welcome to EPG Pathshala. I am Dr. N.P. Singh from Center for Biotechnology, Maharshi Dayanand University, Rohtak. I am going to talk about the module Transcriptomics and Proteomics under the paper Animal Cell Biotechnology. In this particular module, we will going to learn what is the transcriptomic technology, what is the quantitative real-time PCR array, what are the applications of transcriptomics, what is a proteomic technology. Transcriptomics, the study of the complete set of RNAs, transcriptome encoded by the genome of a specific cell or organism at a specific time or under a specific set of conditions. The transcriptome is the total complement of ribonucleic acid, that is RNA, transcripts in a cell and consists of coding, that is 1 to 4% of the messenger, and non-coding, that is more than 95% of the ribosomal transfer, small nuclear, small interfering, micro and long non-coding RNAs. The analysis of mRNAs provides direct insights into cell and tissue specific gene expression features such as presence or absence of transcript, quantification of a transcript, evaluation of alternative or differential splicing to assess or predict protein informs, and quantitative assessment of genotype influence on gene expression using expression quantitative trait loci analysis or allele specific expressions. Contemporary technique in transcriptomics. The dominant contemporary technique in transcriptomics are microarray and RNA sequencing. Microarrays that measure the abundances of a defined set of transcripts via their hybridization to an array of complementary probes. Microarray technology allowed the assay of thousands of transcripts simultaneously. RNA sequencing refers to the sequencing of transcript cDNAs. Massively parallel signature sequencing was an early example based on generating 16 to 20 base pair sequences via a complex series of hybridization. 104 genes of Arabidopsis thaliana were validated through massively parallel signature sequencing in the year 2004. Nowadays, Solexa and Illumina technologies allowed 10 to the power 9 transcript sequences to be recorded. Transcriptomics involves data gathering that includes generating data on RNA transcripts, which can be achieved by either of the two main principles, that is, sequencing of individual transcripts, these are ESTs, and RNA sequencing, or hybridization of transcripts to an ordered arrays of nucleotide probes, these are microarrays. Another important step is data analysis. Transcriptomics methods are highly parallel and require significant computation to produce meaningful data for both microarray and RNA sequencing experiments. Now we will discuss what are microarrays. A microarray is a multiplex, high throughput screening method which uses a two-dimensional arrangement of nucleotide probes on a glass plate or a glass slide, a thin silicon chip. One array can contain 10,000 of nucleotide sequences or hypothetically the complete transcriptome, which is covalently bound to the chip's surface or fixed parallel arose in multifluidic channels. Depending on the scientific questions asked, these nucleotide sequences may represent short fragments, or we can say it as probes with exon sequences to analyze the presence and the quantity of specific mRNA or miRNA in expression profiling experiments. Next generation or deep sequencing, RNA sequencing. RNA sequencing is a new approach to obtain complete information on the RNA expression levels in a tissue sample. RNA sequencing uses a cDNA library which is reverse transcribed from the total RNA in a single tissue or cell culture. These are some different technology platforms used in cDNA sequencing. These are Illumina, Applied Biosystems, Roche Life Sciences, Helicos Biosciences. The sequences retrieved from any of the above technologies are aligned to the reference genome, and the resulting information can be used to analyze both genomic and post-transcriptional mutations and the mRNA expression level of each gene. RNA sequencing therefore extends the information obtained with microarrays by a second dimension, the sequence of the mRNA transcripts. This may permit the identification of disease-associated mutations or SNP in the actively transcribed genes. Another advantage of RNA sequencing is potential analysis of genomes from organisms without complete genomic sequence information. Although further data analysis in these cases is often hampered by the lack of gene annotation or the lack of a sequenced genome. This is in strict contrast to the microarray approach that requires knowledge on the sequences to be detected. Finally, RNA sequences also has a large dynamic range of quantification level, which allows the quantification of very low and high quantities in contrast to the low sensitivity and reduced dynamic range of microarrays. Quantitative real-time PCR arrays. Quantitative real-time PCR arrays are less complex and very valuable method for the quantification of the gene expression of up to 600 genes. Quantitative PCR arrays are a commonly used method for the quantification of the microRNA transcriptome. This is the mainly based on two facts. First, the number of microRNA types per species can be considered at level of hundreds and is therefore much lower than the number of mRNA types. Secondly, the high similarity between different microRNA types makes the development of microarrays difficult due to high risk of cross-hybridization. Now we will discuss what are the application of transcriptomics. Transcriptomics can be applied for the diagnostics or disease profilings. RNA sequencing approaches have allowed for the large-scale identification of transcriptional start sites, uncovered alternative promoter usage, and novel splicing alternatives. These regulatory elements are important in human disease, and therefore defining such variants is crucial to the interpretation of disease association studies. RNA sequencing can also identify disease-associated single nucleotide polymorphisms, that is SNPs, allelic specific expression, and gene fusions, contributing to our understanding of disease causal variants. Retrotransposons are transposable elements which proliferate within eukaryotic genomes through a process involving reverse transcription. RNA sequencing can provide information about the transcription of endogenous retrotransposons that may influence the transcription of neighboring genes by various epigenetic mechanisms that lead to disease. Under Using RNA sequence to understand immune-related disease is expanding rapidly due to the ability to dissect immune cell populations and to sequence T cell and B cell receptors from patients. Next. Human and pathogen transcriptomes. RNA sequences of human pathogens has become an established method quantifying gene expression changes. It also involves identification novel virulence factors, predicting antibiotic resistance, and unveiling host pathogen immune interactions. Responses to the environment. Transcriptomics allows identification of genes and pathways that respond to and counteract biotic and abiotic environmental stresses. The non-targeted nature of transcriptomics allows the identification of transcriptional networks in complex systems. Investigations of gene expression during biofilm formation by the fungal pathogen Candida albicans revealed a co-regulated set of genes critical for biofilm establishment and maintenance. Transcriptomic profiling also provides crucial information on mechanisms of drug resistance. Proteomics. What is proteomics? The word proteome represents the complete protein pool of an organism encoded by the genome. In broader terms, proteomics is defined as total protein content of a cell or that of an organism. The term proteomics was coined in 1997 in analogy with genomics, the study of the genomes. The word proteome is a portmanteau of proteins and genome and was coined by Mark Wilkins in 1994 while he was a PhD student at Macquarie University. Proteomics helps in understanding alteration in protein expression during different stages of life cycle or under a specific stress condition. How proteomics helps in understanding various processes, the structure and function of different proteins. It also helps us to understand the protein-protein interaction of an organism. A major defect in either protein structure, its function, or alteration in expression pattern can be easily detected using these proteomic studies. This is important with regard to drug development and understanding various biological processes, as proteins are the most favorable targets for various drugs. Proteomics can be divided into functional proteomics, structural proteomics, and differential proteomics. Functional proteomics is the identification of protein-protein, protein DNA, and protein RNA interaction which affects the function of the cell. Structural proteomics is the identification of all interactions by metal ions, toxins, drugs, etc., which affecting the protein structure. Differential proteomics, determination of differences in protein expression under a particular stress conditions. Various techniques involved in proteomic studies. In proteomics, there are multiple methods to study proteins. Generally, proteins can either be detected using antibodies or using mass spectrometry. If a complex biological sample is analyzed, either a very specific antibody needs to be used in quantitative dot plot analysis, or then biochemical separation needs to be used before the detection step, as there are too many analytes in the sample to perform accurate detection and quantification. Now, how these proteins can be detected with the help of antibodies? We can say it as immunoassays. Antibodies to particular proteins or to their modified forms have been used in biochemistry and cell biology studies. The enzyme-linked immunosorbent assay, that is ELISA, has been used for decades to detect and quantitatively measure proteins in the sample. The Western blot can be used for detection and quantification of individual proteins, where in an initial step a complex protein mixture is separated using SDS page, and then the protein of interest is identified by using an antibody. Antibody-free protein detection. Antibody-free protein detection offers various advantages, often able to determine the sequence of a protein or peptide. They may have higher throughput than antibody-based, and then they sometimes can identify and quantify proteins for which no body exists. Detection methods. One of the earliest methods for protein analysis is Edmund degradation, which was introduced in 1967, where a single peptide is subjected to multiple steps of chemical degradation to resolve its sequence. Recent methods use mass spectrometry-based techniques based on soft ionization methods such as matrix-assisted laser deionization and electrospray ionization methods. The separation methods for proteomics. For the analysis of complex biological samples, a reduction of sample complexity is required. This can be performed offline by one-dimensional or two-dimensional gel electrophoresis separation. More recently, online methods have been developed where individual peptides in bottom-to-up proteomics approach are separated using reverse phase chromatography and then directly ionized using electrospray ionization. The direct coupling of separation and analysis explains the terms online analysis. In two-dimensional gel electrophoresis, we will have to run our proteins firstly in one-dimensional gel electrophoresis using isoelectric focusing gels in play, which is later on placed on the SDS polyacrylamide gels for the second dimensional gel electrophoresis. During the isoelectric focusing, the proteins will be separated on the basis of their isoelectric points, and these proteins will be further run on the second gel, which is based on the molecular weight. In one-dimensional gel electrophoresis, the proteins will be separated on the basis of their isoelectric points, and on the second dimension, these will be run or these will be separated on the basis of their molecular weights. Now we will discuss how throughput proteomic technologies can be used for biological studies. Proteomics has steadily gained momentum over a past decade with the evolution of several approaches. Few of these are new and others build on traditional methods, mass spectrometry based methods, and microarrays are the most common technologies for large-scale studies of proteins. Mass spectrometry and protein profiling. There are two mass spectrometric-based methods which are currently used for protein profiling. Two-dimensional gel electrophoresis to separate proteins from different samples in parallel, followed by selection and staining of the differentially expressed proteins to be identified by mass spectrometry. Here we are discussing how the proteins will be studied for the identification of those proteins. Firstly, we will have to isolate that protein from the sample, and then we will run it on first dimensional gel electrophoresis. Later on, that particular gel will be run on the second dimension for the complete separation of those proteins, both on the basis of their isoelectric points as well as on the basis of their molecular weights. These unidentified proteins, which later on extracted from the gel, and these proteins which has been extracted from the gels will be later on digested by the proteases. These proteases cuts those proteins into the small peptides, or they will digest it into the small peptides, and these peptides will be studied by using mass spectrometry methods, and these mass spectrometry methods, they will determine the mass of those peptides. The output of the mass spectrometric method, which will determine the masses of various peptides, is in the form of peptide mass fingerprinting. In peptide mass fingerprinting, as we can see in the picture, we can have various masses of those peptides in a graphical way, and after the determination of those masses, we can determine the amino acid sequences, and these amino acid sequences can be comprised with the database, which are included large number of protein in the databases. After the determination of amino acid sequence from the peptide mass fingerprint, we can compare these sequences from the databases which has already been submitted. The databases such like Uniprot, and from there we can identify our unidentified proteins, and we can also know their functions which has already been submitted in the databases. The second quantitative approach uses stable isotope tag to differentially label proteins from two different complex mixtures. Here, the cysteine residues of proteins within a complex mixtures are labeled first isotopically with isotope coded affinity tags reagents, and then digested to yield labeled peptides. The labeled mixtures are then combined, the peptides separated by multidimensional liquid chromatography and analyzed by tandem mass spectrometry. We will discuss what are the applications of proteomics. Proteomics are broad applications in all the aspects of life sciences, including several practical applications as drug development against several diseases, differences in expression protein pattern profiling of normal and diseased person by analyzed the target proteins. Protein to gene may be predicted. Once protein and gene is identified, functions may be predicted. This can be done with the help in the disease management and drug development strategies. In this figure, we can compare two different techniques. These are transcriptomics and proteomics. How these two techniques are different. In case of transcriptomics, we can take tissues from the various treated samples, and we will isolate the RNA, and after isolating this RNA, we will pre-processing for microarrays. After hybridization and scanning, we will get the differentially expressed genes which represent the transcript profiling of that particular tissues. In case of proteomics, we will take up the tissue and then isolate that proteins and leads to the tryptic digestion, and after tryptic digestion, these tryptic peptides will be treated in high-track labeled peptides and samples. The two-dimensional fractionation of peptides using reverse phase chromatography and loaded these fractions on the MALDI top, and after that we will pre-processing and analyze using protein pilots. After normalization, these proteins will be studied for their differential expression, and ultimately we will get a proteome profiling of those particular differentially expressed tissues. So students, in this particular module, we learned transcriptome and proteome analysis have been introduced as a research tool in most fields of molecular biology research. They permit the identification of biomarkers, gene expression profiles, and the understanding of complex molecular mechanisms in cell physiology. Methods to analyze the transcriptome have made greatest advancements so far. cDNA microarray have been and are widely used to analyze disease mechanisms and to identify prognostic gene sets. Proteome analysis is considered as an ultimate method for the analysis of gene expression and function mechanism. 2D gel electrophoresis allows protein separation with less expensive equipment and experience. Chromatography-based proteomics seems to be of greater development potential in terms of mechanization and standardization. The analysis of chemically and biochemically high diverse proteins when in tissue lysates, that is to cover the full proteome in each case. Thank you.